Good morning.
Good morning.
Non-PCR.
morning.
Good morning. Okay. Non-PCR amplification techniques. This is our lecture today. Previously, you have heard about PCR reaction, about types of PCR, so you already know that the PCR is still the go-to method in the molecular diagnostics. But with the rise of close to patient testing, non-PCR amplification methods that are also called isothermal amplification methods, with their low power and potentially easier detection schemes, look increasingly attractive. Isothermal amplification techniques of amplification of nucleic acids, they have emerged as an alternative in which rapid and efficient amplification is achieved at a constant temperature without the thermal cycling that is required in traditional PCR reaction. Additionally, isothermal amplification can be performed under simple conditions. So, for example, in a water bath, on the cell surface, or even inside the living cells, all of which are not feasible with PCR reaction. And since the early 90s, many isothermal nucleic acid amplification techniques employing different amplification mechanisms have been developed. And most of these methods possesses impressive sensitivity for the detection of nucleic acids, and some have achieved also commercial success. More recently, isothermal amplification techniques have been expanded to detect a wide spectrum of targets, including proteins, cells, small molecules, and also ions. The combination of isothermal amplification and nanotechnology has also attracted intense attention. Isothermal amplification provides abundant nucleic acids as materials for constructing higher order nucleic acids, nanostructures, nucleic acid templated metal nanostructures, and nucleic acid hydrogels, which hold a really great promise in biosensing, bioimaging, and nanomedicine. Also, real-time readings of amplification are possible with these methods, and amplified products detected by measuring turbidity or by visual inspection for color change. This capability eliminates the need for gel electrophoresis. These isothermal amplification methods could become more popular than PCR soon, owing to their low energy requirement and simplicity, which allows for their possible integration into simple compact systems. Of course, isothermal amplification methods, they differ from one another in features such as the number of primers and enzymes that are used, or the temperature of reaction. Transcription-based amplification includes transcription-mediated amplification and nucleic acid sequence-based amplification, so TMA and NASBA. Both methods are isothermal amplification reactions mimicking retroviral RNA replication. Both are specific for target RNA sequences and have been gaining popularity because they have been shown to have a wide range of applications for pathogen detection in clinical, environmental, and food samples. Both methods are also convenient to use with commercially available kits. Transcription-mediated amplification involves the isothermal, of course, amplification of rRNA by reverse transcription and subsequent generation of numerous transcripts by RNA polymerase. And following amplification, these RNA copies are hybridized with a complementary oligonucleotide probe for detection via chemiluminescent tag. This method may produce up to 1,000 copies per cycle, resulting around 10 billion fold increase within around 30 minutes. And once RNA is present in this reaction, the first primer attaches to its complementary site at the 3' end on the template. The reverse transcriptase enzyme is used to make the complementary DNA, so the cDNA of the RNA target. RNase H then degrades the initial RNA template only with RNA-DNA hybrids, but not single-stranded RNA. The second primer attaches to the 5' end of the DNA strand and is extended to form a double-stranded DNA molecule using reverse transcriptase polymerase activity. The T7 RNA polymerase promoter is incorporated in the first primer, enabling RNA polymerase to produce a complementary negative sense RNA, so the amplicon, which can be used again in the initial step of the reaction. And the process repeats automatically, resulting in an exponential amplification of the original target that can produce over 1 billion copies of amplification in less than 30 minutes. The original procedure had the disadvantage that a heating step was required to denature the intermediate RNA-DNA hybrid product. The heat also denatured the enzyme, so the fresh enzyme had to be added after each denaturation step, and the process was simplified with the addition of RNase H derived from E. coli. This method, transcription-based transcription-mediated amplification, is popular in clinical diagnostics, with many commercial tests based on the technique available to clinical laboratories. Targeted RNA allows for the direct detection of RNA viruses, so HCV, HIV. This technique has also been investigated by labs and research groups that are concerned with food and water safety. Nucleic acid sequence-based amplification is also known as self-sustained sequence replication. It is similar to TMA method. NASBA requires the three enzymes, so reverse transcriptase, RNase H, and DNA-dependent RNA polymerase, whereas TMA utilizes a unique reverse transcriptase with internal RNase H activity. The principle of this method is illustrated here, and during the reaction, the target RNA first hybridizes to the forward primer and is converted to a complementary DNA intermediate by reverse transcriptase and RNase H. And then this intermediate double-stranded cDNA that contains a promoter region is formed using the second primer, and many antisense RNA strands to the target RNA are produced by transcription of this cDNA. And they become templates for the original reverse transcription reaction. And the newly synthesized antisense RNA and cDNA, they serve as templates for continuous cycling of the reverse transcription reaction, resulting in exponential accumulation of antisense RNA that is complementary to the target RNA. Generally, NASBA is very sensitive. Its sensitivity is maybe is similar to that of real-time PCR reaction. Another advantage of this method is that the amplified product is single-stranded RNA, enabling simple and rapid hybridization detection without denaturation step. And many assay approaches, such as gel electrophoresis, real-time fluorescence, or colorimetric assays, have been used to detect the NASBA amplicon. Also, multiplex detection by NASBA was demonstrated. Sometimes an additional step to remove the secondary structure of target RNA and for primer annealing at 65 Celsius degrees is required before the amplification reaction. And both the amplification and detection processes, they require RNase-free conditions to prevent RNA degradation. Strand displacement amplification. This is of course, isothermal amplification technique that it was described in 1992, so it's not so new. It relies upon the activities of a restriction endonuclease and the exonuclease deficient DNA polymerase to continually amplify a DNA target. Here, in this method, inner primers are designed to have a restriction enzyme recognition sequence within their 5' overhang. Flanking outer primers bind upstream of the inner primer targets. And extension of all 4 primers by DNA polymerase results in amplicon incorporating restriction enzyme sites. These steps you can see on a picture. And the restriction endonuclease added to the reaction creates a single-stranded nick at these sites, where DNA polymerase can then initiate replication and displace the annealed strand. And the polymerase that is used in strand displacement amplification must be able to initiate polymerization at a nick site and must lack exonuclease activity. Such polymerases include exoclenov or exo-BST polymerases. The restriction endonuclease must be able to create a single-stranded nick at a recognition site. It must also dissociate quickly to allow the polymerase to act and perform rounds of nicking and dissociation. And the maximum target length is influenced in this method by different reaction conditions. For example, the time of the DNA polymerase, the frequency of recognition sites of the restriction enzyme surrounding the target sequence. Generally, this method is used for short fragments between 50 to 120 base pairs long, but this method is not well for longer fragments than 150 and 50 base pairs. Very important non-PCR method is loop-mediated isothermal amplification. This method requires a set of 4 specifically designed primers that can recognize 6 distinct regions on the target DNA and DNA polymerase with strand displacement activity. This method can amplify a target 1 billion times in less than 1 hour. The amplification products are stem loop DNA structures with several inverted repeats of the target and cauliflower-like structures with multiple loops. By combining processes for simplicity, rapidity, and precision, this LAMP method could be employed in a wide range of applications. So in point-of-care testing, genetic testing, in clinical settings, and rapid testing of food products and environmental samples. The LAMP method can also amplify RNA with the addition of reverse transcriptase. The synthesis of large amounts of DNA in a short time creates the byproduct pyrophosphate, which is produced during the reaction and forms a white precipitate that can be used to detect positive reactions, both visually by observing the precipitate and quantitatively by measuring the turbidity change for real-time analysis. Loop-mediated isothermal amplification can amplify a few copies of the target in less than 1 hour, even when large amounts of non-target DNA are present. And this method has been applied to detect different viral pathogens, including dengue or SARS virus, and also highly pathogenic avian influenza viruses. Several aspects of this method reaction differ from those of other amplification methods. First, only a single type of enzyme is required here, and the amplification can be carried out at a constant temperature. Also, LAMP uses 6 gene regions for amplification, and the fundamental characteristics of the inner primer provide the amplification with a specificity that is much greater than that observed in other methods. The amplification process is usually completed within 1 hour with sensitivity similar to that of nested PCR reaction. The rapid progression of the LAMP amplification reaction contributes to its high amplification efficiency. Also, as I said before, small quantities of a gene can be amplified with a short time. This method can be also used to amplify the target RNA sequence. In this case, one-stem amplification, the same as the amplification of DNA, can be performed by the addition of a reverse transcriptase enzyme, because the reverse transcriptase also exhibits strand displacement activity. Simple detection methods such as turbidity or fluorescence can be employed because of the high specificity and large output of the amplification products. The turbidity detection method uses the turbidity of magnesium pyrophosphate, that is a byproduct of DNA synthesis, as an indicator, whereas the fluorescence detection method uses fluorescent chelation reagents. Both methods allow for real-time detection and visual inspection. The two probes, oligonucleotide probes that are used in this assay, bind to the target in a juxtaposed manner. But upon target binding, a structure-specific flap is created at the junction where the two probes meet. And this flap can be cleaved by a flap endonuclease. So if a fluorophore and quencher are positioned around the cleavage site, then the separation of the fluorophore and quencher would generate fluorescence, and the fluorophore labeled probe is designed to have a melting temperature close to the assay temperature to enable the probe to frequently bind and detach. And when the probe binds, the flap endonuclease cleaves it, and after detachment, a new intact fluorescently labeled probe anneals to the same site. It has been estimated that for one target, about 3,000 cleavages can be done in about 90 minutes. Although this amplification rate is quite good, it is not enough for detection of low copy number targets, and so for low analyte concentration, an initial PCR amplification of the target is necessary before the invader assay. The sequence at the overlap region is important in invader assay for effective cleavage by flap endonuclease. The invader assay contains two oligonucleotide probes which can hybridize to the target DNA. And when bound to target DNA, a three-dimensional invader structure is formed, which is recognized by flap endonucleases. The cleavage reaction by flap endonucleases generates fluorescent output signal. And when the probe does not match the allele present in the target, there will be no overlapping invader structure, so the probe cannot be cleaved and there will be no fluorescence signal. A second generation invader assay combines the two invader cleavage reactions into a single assay. The first cleavage reaction is similar to the original invader assay strategy. However, after the first cleavage step, the flap sequence, once released, acts as an invader oligonucleotide and anneals to another thread cassette, which can be cleaved by flap endonuclease. The invader assay has been used to detect the factor V gene, for example, or genes responsible for hearing loss or for detection of human papilloma virus. Another interesting method is rolling circle amplification. This method uses strand displacing DNA polymerase to continuously amplify a circular DNA template at a constant low temperature, producing a long DNA molecule with tandem repeats of the circular template. This reaction, RCA, involves many rounds of isothermal enzymatic synthesis in which DNA polymerase extends a circle hybridized single-stranded DNA primer by continuously progressing around the circular DNA probe of several dozen nucleotides to replicate its sequence over and over again. And after completing DNA synthesis in the primer of the strand displacement ability, continues to synthesize a new strand by displacing the previously formed double-stranded DNA, and finally produces long single-stranded DNA amplicon that possesses repeated sequence of the original target. And this reaction is widely used for diagnostic purposes in direct or indirect detection of different nucleic acids, protein, and other biomarkers. Rolling circle amplification can be coupled to the ligation of a padlock probe to provide sensitive detection of nucleic acids. And the padlock probe is a single probe that contains 3' and 5' and sequences that bind to the target sequence in a juxtaposed manner to aid a ligase-mediated circularization of the padlock probe. After this circularization of the padlock probe, a primer and DNA polymerase, they are used to produce these long repeating DNA sequences, which can be then detected via a variety of methods. And this padlock probe has been used to detect several disease-related genes such as cystic fibrosis, transmembrane concatenance regulator that leads to cystic fibrosis, detection of Epstein-Barr virus, or detection of influenza virus mutations. The target sequence recycled rolling circle amplification. It is obviously based on RCA method, but does not utilize ligases as is done in padlock probe. In the original RCA technique, the target facilitates the circularization of the probe, which is then used as a template for PCR. But here for this TR-RCA, the circular DNA template is required for the amplified sensing. Is already circularized, but the primer binding site is sequestered in the duplex region of the dumbbell. And upon target binding, the duplex region opens up opens up to enable the primers to bind for the RCA process to begin. So for this TR-RCA method, target binds to dumbbell probe to open the dumbbell structure into a circular template. So it's step 1 on the picture, and then the circular template can then be amplified via the regular RCA reaction. Hyperbranched RCA is another improvement of RCA reaction, and here to increase the sensitivity of RCA, both forward and reverse primers are used. So during this hyperbranch RCA, at first forward primers binds to the circular probe and is extended by DNA polymerase. After generation of a long single-stranded DNA that contains binding sites for the reverse primer, the reverse primer binds to this product and is extended by DNA polymerase. And the nascent single-stranded DNA generated from reverse primer extension can serve as a template to synthesize new DNA. The SMART technology, so signal-mediated amplification of RNA technology, is based on the formation of the three-way junction structure. It consists of 2 single-stranded oligonucleotide probes. Each probe includes one region that can hybridize to the targets at adjacent positions, and other much shorter region that hybridizes to the other probe. And the 2 probes are annealed to each other in the presence of the specific targets and forms a three-way junction. And following this three-way junction formation, DNA polymerase extends the short probe by copying the opposing template probe to produce a double-stranded RNA polymerase promoter sequence. And this formed promoter allows RNA polymerase to generate multiple copies of an RNA amplicon, and therefore being produced only when a specific target is presented to allow for this three-way junction formation. Each RNA amplicon may itself be amplified by binding to a second template oligonucleotide and is extended by DNA polymerase to generate a double-stranded promoter. This leads to transcription that increases the RNA amplicons, and the RNA amplicons can be detected by an enzyme-linked oligosorbent assay or in real-time format. This method actually relies on a signal amplification and does not require thermal cycling or involve the copying of target sequences. The assay generates a signal that is highly target dependent and is appropriate for the detection of DNA and RNA targets. Isothermal multiple displacement amplification. This is also based on strand displacement replication, but by multiple primers. In this method, 2 sets of primers are used, right set and left set of primers. The primers in the right set are complementary to one strand of the nucleic acid molecule to be amplified, and the primers in the left set are of course complementary to the opposite strand, and the 5' ends of primers in both sets are distal to the nucleic acid sequence of interest when the primers have hybridized to the nucleic acid sequence molecule that is amplified. And the amplification proceeds by replication initiated at each primer and continuing through the nucleic acid sequence of interest. A key feature of this method is the displacement of intervening primers during replication by the polymerase. And this method has been further developed for whole genome amplification by using a random set of primers to randomly prime a sample of genomic nucleic acid. And in this way, multiple overlapping copies of the entire genome can be synthesized in a short time. Here you can see an example of isothermal multiple strand displacement amplification. At the top, there is a double-stranded nucleic acid molecule which contains nucleic acid of interest, the hatched area. Hybridized to the nucleic acid molecules are right and left sets of primers, and the middle panel is the multiple strands of replicated nucleic acid being elongated from each primer. The polymerase at the end of each elongating strand displaces the elongating strand of the primer ahead of it. And the bottom panel is the multiple strands of replicated nucleic acids further elongated. Also, here are shown the next sets of primers which hybridize to their complementary sites on the newly replicated strands. The newly replicated strands are made available for hybridization to the primers through displacement by the polymerase elongating the following strand. Helicase-dependent amplification. This is a method that is based on the natural mechanism of the DNA replication fork, and it employs DNA helicase to denature double-stranded DNA instead of conventional heat denaturation step that is used in PCR reaction. The separated nucleotides by helicase are protected by single-stranded binding protein, allowing the binding of primers to initiate elongation by polymerase. Of course, at a constant temperature, without any thermal steps, unlike both SDA and RCA methods, which require an initial heat denaturation step. Even through the rest of the amplification reaction is isothermal. And the newly synthesized double-stranded DNA products are then used as a substrate for the next round of the reaction, which results in exponential amplification of the target sequence. The simple reaction, when also is combined with other advancements such as the ability to detect RNA with reverse transcription, gives this helicase-dependent amplification much potential for use in portable nucleic acid detection system. Recombinase polymerase amplification uses of course, the recombinase, that is a protein which catalyzes the hybridization process of short primers, oligonucleotide primers, with the complementary target sequence. And during this reaction, the recombinase primer complexes initiate the amplification cycle by scanning the duplex template for specific sequences. And once the specific site is found, the enzyme opens the double strands to allow for the hybridization of the primer and target sequence. And this process is aided by single-stranded DNA binding proteins, which bind to the this melted DNA. The primer is then elongated with the newly synthesized strand displacing the O strand, and the newly synthesized duplex can then act as a template for the next cycle. This method is fast and sensitive, but the background noise can be an issue, and to eliminate the background noise, modified primer that contains a cleavage site for the double-strand specific E. coli endonuclease 4 is usually used. The primer can be also labeled with fluorophore and quencher so that the cleavage reaction is accompanied by increase of fluorescence, and the nuclease step in this reaction serves as an additional proofreading step to eliminate background noise. The junction or Y probes, they are also used in isothermal amplification. Restriction endonucleases cleave duplex DNA to form either a blunt end or a sticky end. But one of the disadvantages of using them for nucleic acid diagnostics is they need to have a specific recognition site in the target analyte. And it has been demonstrated that this requirement to have the recognition site in the target sequence can be obviated when probes that form this Y or junction structures are used. And in this Y probe junction probe strategy, two probes that have limited complementarity, usually between 4 to 6 base pairs, they do not bind to each other to an appreciable extent, but when an analyte that binds to the two probes in a juxtaposed manner is present, this Y structure is formed. So if the segment of complementarity between the two probes contains a restriction endonuclease recognition site, then the formation of this Y structure would lead to restriction endonuclease cleavage, and the process can be repeated, leading to amplified sensing under isothermal conditions. In the last few years, many modifications of the junction probe platform have been reported. For example, it has been shown that the incorporation of the phosphorothioate moiety in one of the probes that is called the helper probe results in nicking scenario and more sensitive detection. The different isothermal detection strategies that have been developed over the years are they are being to be put into use for the detection of nucleic acids from biological samples. One of the next methods demonstrated an ultra-sensitive detection of microRNA using parallel amplification cycles involving polymerase extension, nicking endonuclease cleavage, and exonuclease-mediated target recycling. And this detection assay could be used to detect microRNAs in crude extracts of cancer cells. It has been also demonstrated that catalytic hairpin assemble can be combined with several isothermal amplification methods, including LAMP, SDA, and RCA, to provide a more sensitive detection of nucleic acid. It was also pointed out that catalytic hairpin assemble is adaptable to different temperatures, buffers enzymes and detection, and therefore holds promise for improving upon many of the isothermal detection strategies developed today. One of these parallel isothermal detection strategies is called split peroxidase-like DNA enzyme probes. This method uses hemine binding quadruplex DNA peroxidase. And here two probes contain two regions, the analyte binding arms and the split peroxidase parts. And the splitting pattern can be even or uneven, and when the two probes are separate, they show no or very low peroxidase activity. When the target hybridized to both of the two probes, the G quadruplex peroxidase-like DNA enzyme is assembled. It has been reported that the introduction of a stem loop or loop-stem loop motif connecting the G-feed tracts can effectively reduce the background noise, and sub-nanomolar targets could be detected. And this split peroxidase-like DNA enzyme probes assays were applied for the detection of PCR amplified DNA from Salmonella or Mycobacterium. This assay is fast and convenient, but it's also limited by the detection of nanomolar analyte. Several strategies for DNA or RNA detection use either ribozymes or RNA cleaving DNA enzymes. These methods include catalytic molecular beacon strategy and detection of RNA using hairpin ribozymes. In the first catalytic molecular beacon strategy, by binding of the target gene, inactivated stem loop molecular beacon probe is opened, and fluorescent label substrate is cleaved, resulting in a change in the fluorescence profile. Detection of microRNA using hairpin ribozyme. Here the domain C is the binding site for the target's microRNA. So the LED7. In the presence of this LED7, the hairpin ribozyme is opened up and the probe substrate is cleaved. Another methods in this group are called target-assisted self-cleavage and also binary deoxyribosome probes. In this target-assisted self-cleavage, the probe contains the deoxyribosomes moiety, which can catalytically cleave RNA, and the probe contains target binding arms and is labeled with fluorophore and a quencher, separated by a short nucleic acid sequence embedding a ribonucleotide phosphodiester bond. And in binary, the deoxyribosome probes that use the T-hype strategy. Here two probes are used which have analyte binding arms and substrate binding arms. In the presence of analyte, the two probes bind to the analyte to form a tripartite structure, which is an active DNA enzyme, and the DNA enzyme can then cleave a fluorogenic oligonucleotide containing this ribonucleotide cleavage site. The preceding enzyme-mediated isothermal detection of DNA or RNA methods are not perfect. One of the limitations of these methods that are based on enzymes is that sample quality and the presence of other biomolecules can affect the sensitivity of the method. Also, the adaptation of the strategies for detecting nucleic acids in leaf cells is not trivial. So therefore, some efforts have been aimed at developing non-enzymatic reactions, and many types of DNA template catalyzed reactions for detection purposes have emerged in the last 20 years. And these approaches are divided into two types: chemical ligation-based methods and ligation-free detection. And quenched autoligation, fluorescence energy transfer, and fluorophore reconstitution are two classic examples of the detection of nucleic acids using ligation-based methods, whereas template-assisted fluorophore generation and reporter transfer reaction represent ligation-free methods. In the quench autoligation fluorescence energy transfer strategy, the fluorophore on one of the probes is quenched by a dapsyl group, which can act as a living group. And after a template catalyzed ligation of two probes, the dapsyl quencher becomes separated from the fan fluorophore, leading to increase of the fluorescence. In a fluorophore reconstitution method, the non-fluorescent indolinium and aldehyde-labeled complementary DNAs reacts to form a hemicyanine catalyzed by diamine, and the ligated reaction product is fluorescent. In reported transfer reaction, at first quencher transfer reaction is performed and then catalytic cycle of template catalyzed quencher transfer is done, and the probes here can react with each other to generate a signal, but the reaction is significantly increased when the two probes bind to the same targets. Non-covalent DNA catalytic reaction uses an input signal to catalyze the release of an oligonucleotide that can be used to catalyze the next reaction. These methods have also been used to detect nucleic acid. In most cases, the strength displacement begins after the target nucleic acid analyte has bound to the toehold segment of the initiation probe. In multiple strength displacement via this non-covalent DNA catalysis, for metastable probes A, B, C, and D are used. And probe A can form a stable complex with probe B, but there is a kinetic barrier to this complex formation, and the target can bind to probe A to open the stem loop, and this facilitates the binding of probe A to probe B. So then the probe AB complex has an overhang which can bind to probe C to open up the stem loop structure in probe C. Then the probe ABC complex contains an overhang that can bind to the probe D to open the stem loop structure of probe D to form a transient probe ABCD complex. Because probes D and B compete for binding to the same site of the probe C, the binding of probe D to the ABC complex result in the dissociation of AB complex, which can initiate another round of probe CD opening. Probe D is designed to also contain a region of complementarity to probe A, so that the probe CD complex can also open the stem loop structure of probe A. And this series of cascade reaction lead to exponential amplification The last isothermal amplification that I would like to present is hybridization chain reaction. This is a process where a target DNA or RNA can initiate the assembly of long double-stranded DNA structures, which contain nicked regions. Probes that are used for this method are usually in a hairpin form, so they do not associate with each other. However, in the presence of a target analyte, one of the stem loop probes is opened up, and this initiates a cascade process which opens up other hairpin probe to form a long alternating double-stranded DNA structure. And by incorporating fluorescent turn-on probes or latent DNA enzymes into the hairpin probe, this reaction process can lead to amplified sensing. In this hybridization chain reaction, two molecular beacon probes that are shown in a picture can hybridize with each other only after the hairpin structure of one of the probes, that is colored black, is open upon target binding. The target gene binds to reaction binds to the region A in the first probe to open up the stem loop structure. This is the initiation step. Then the complex of target gene and the first probe binds to C region in the second probe, and the second probe is opened up. And then complex containing the target and two probes binds region A in the probe 1 and continue this hybridization chain reaction. So far, a number of nucleic acid detection methods that operate under isothermal conditions have been described. Some of them have been shown to detect few copy numbers of nucleic acids and hold promise for point-of-care diagnostics. Detection strategies that utilize the tandem or parallel amplification methods could allow for ultra-sensitive detection of analytes under isothermal conditions, but these strategies utilize many enzymes and reagents, such as nucleotides, and could prove to be expensive. Moreover, the use of many enzymes in detection assay brings into play other problems, such as enzyme storage, and they need to find assay conditions that are permissive for all of the enzymes being used. Most DNA modifying enzymes require divalent cations for efficient catalysis, but the optimal concentration of cations are not necessarily the same for all enzymes, so it's almost impossible to use specific cations concentrations that are optimal for all of the enzymes in parallel catalysis. In some instances, enzyme being used in parallel reactions might require different additives or temperatures for optimal performance. And in some cases, an additive that increases the catalysis of one enzyme might inhibit another enzyme, making the optimization of this tandem reactions not so trivial. So why isothermal amplification is not as popular as PCR PCR reaction. PCR came just a little bit before isothermal amplification, and the principle behind it was relatively easy to understand. At programmed temperature intervals, the behavior of DNA primers and polymerase could be changed into exponential amplification. Because it was first and quite easy, it was quickly adopted by laboratories and isothermal amplification. It's much more complicated in designing primers. There are more components that are needed depending on the method. Still, most of these methods are quite difficult to understand, and also PCR reaction is much faster than isothermal amplification. The key difference between isothermal amplification and PCR reaction is that the isothermal amplification is carried out at a constant temperature using amplification, whereas PCR requires temperature changing cycle for amplification. What this also means that isothermal amplification does not require a thermal cycler for the reaction. Another important difference between PCR and isothermal amplification is that DNA polymerase will extend primers on a single strand of DNA during PCR, while certain isothermal amplification techniques have special strand displacing DNA polymerases that extend primers on double-stranded. What you should know from this lecture is to know and understand different types of this isothermal amplification methods, especially LAMP amplification, TMA, NASBA, SDA, and RCA methods, and to know the differences between these methods. Also, you should know the differences between isothermal amplification and PCR reaction, and I can recommend a review article for you that has all the isothermal amplification, but I would like you to focus on the mostly known methods. You don't need to know all the methods are described in this paper, just the most widely known. And this is for today. Thank you for your attention.
Thank you, Sybil. Thank you. Goodbye.
Thank you. Bye.
Thank you. Goodbye.
Goodbye.
Thank you. Bye.
Dzień dobry pani doktor. Ja się chciałam zapytać, czy może doszedł już ten mail ode mnie?